Today I'll be showing you how to use KnockTF. KnockTF is a secondary, manually annotated database that contains information about human transcription factors and their effects on gene expression. The database consists of 570 transcription factor (TF) datasets for 308 different TFs. These datasets contain data on differential expression of human genes under knockout and knockdown of a given TF, showing the effects of a TF on a given tissue sample or cell type. It also provides functional annotation and analysis on target genes and information about TF interactions on gene promoters and enhancers. Browsing gives an idea of the KnockTF data. Here you can get a breakdown of the data sources, sample types, tissue types, transcription factor superclasses, and individual transcription factors contained in KnockTF. You can also filter datasets by these topics by clicking them. For instance, if I wanted to see datasets for bone marrow, I could click here, giving me a list of the 13 datasets generated from bone marrow. Search by TF allows you to find data on a specific TF or a type of TF. You can search by TF class or superclass, or you can look up a specific TF. This section visualizes all of the TFs in the database by superclass. If I search for the REST TF, I get 5 datasets from different tissue sources. If I want to look at REST expression in the adrenal gland, I can see an overview of REST, including the knock method of this dataset, the sequence REST targets on the DNA, its TF classification, and some information about the sample this data is from. I can also see a network of the top 100 differentially expressed genes in this set, with upregulated genes in red and downregulated genes in blue. I can change the parameters of this graph to see less genes, or only up or downregulated genes. Here is a list of differentially expressed genes in this dataset. The default sorting here is by rank, which is determined by fold change. This table also provides a list of TFs that alter the expression of each target gene, sorted by predicted interactions with promoters, superenhancers, and typical enhancers of the gene, with interactions supported by ChIP-seq data in red. Next is functional analysis of the target genes, with KEGG pathway, Go enrichment, and Gene Set Enrichment Analysis. This gives information about potential effects REST expression could have on target cell. Upstream annotation links are provided for further information about conditions related to REST. A heatmap with a dendrogram and a scatterplot show the differential expression of the target genes. The number of genes in the heatmap can be adjusted. The Expression Atlas displays data for expression levels of REST in normal human tissue, cancers, and human and cancerous cell lines. Search by knock type and tissue type work very similarly to search by TF and allow us to refine our search to a functional class of TFs or a certain sample type. You can also search by target gene. Let's look at SCG3. We can search for it by its gene symbol, entree ID, or its ensemble ID. I'll use gene symbol. We can also adjust the fold change level of our search. Here we get a list of TF datasets where SCG3 is differentially expressed. Or we can click here to get an overview of SCG3. This provides information about SCG3 and a word map of the TFs that affect its expression, with blue for repression and orange for activation. Color intensity indicates the level of expression change. We can also get the FC and log2 FC by hovering over a given TF. SCG3 TF pairs is a table of all of the TFs in the database that affect SCG3 expression, with information on sample sources, fold change values, and p-values. Regulation region predicts how TFs affect SCG3 expression, with identified and predicted data for promoters and 2 types of enhancers. Red boxes indicate a significant experimental and predicted regulatory effect by that TF. The Expression Atlas is the same as for the TFs, but for the target genes. Some issues with KnockTF include broken links, like the upstream pathway annotation links leading to a 504 timeout error. The website also provides analysis options to generate subnetworks of transcription factors and target genes, and further enrichment of transcription factors. However, neither of these appear to work, as the site provided examples yield no results. Apart from these issues, The database is well designed and runs very quickly, while providing useful information on human transcription factors. It is well suited to its task, however it is rather limited in its scope. There are plans to add data on other organisms and more human datasets in the future, which would make this database more widely applicable as transcription factors play a larger role in how we look at gene expression.